All right. And I'm going to be letting people into the seminar while I'm just doing this little bit of intro. So we are recording today, so everybody knows about that. We have the talk schedule is posted at innovatebio.org. You can find it right in the front page. You look under events. And I want to tell you a little bit about our goals for the series. Our goal for the series here is to showcase a lot of biotech-related ATE projects. The ATE projects are all projects that are funded by the National Science Foundation's Advanced Technological Education Program, which is a really wonderful thing and one of the best kept secrets, I understand. So we want everybody to know about it. We want people to understand what's involved with the grants, what people are doing. And we want you to know how to talk and connect to each other so that you can learn from each other. So next week, we're going to be having a talk from a group that is in the progress of applying for an AT grant, a consortium grant. They don't have it yet, but they're working on it, and they're going to show you what they're doing, give you a peek behind the curtains. And that one is going to be covering developing the core technology workforce. It's not always clear to everybody, but the biggest employers of biotechnicians are actually universities in many places. So how do we get students into those jobs, and how can we work with the universities to change their hiring practices to make sure that they hire community college graduates? That's going to be a big focus of that grant and that project, developing those research careers related to biotechnology. So today, for our format, we're going to have the talks will be about 20 minutes each. We're going to have a discussion and questions and evaluation at the end. We can continue the discussion if you'd like. I put an invitation to join Slack into the chat, so you can continue that there. And the discussion will be in the ATE project channel once you've joined. And now, without further ado, I'm going to introduce our first speaker, Dr. John McDowell, who is from the science department at Delaware Technical Community College. And he is going to be talking about technician training in CRISPR gene editing. So I'm going to stop sharing my screen. And John, it's up to you now.
Okay, let's hope everything goes well. So Sandy, thanks for that introduction. I don't know about anybody else, but I seem to have gotten the Zoom yips during a class yesterday. My Zoom connection blipped out for like 2 or 3 seconds at a time over the course of the lecture. So hopefully everything will go smoothly with today. But thank you to everybody at Innovate Bio, Sandy, Linnea, and the group for having the opportunity to talk about our project. I'm very excited about this chance and what we're doing in the training of CRISPR gene editing. I figured today I would give a little bit of background of our college and especially the programs that the curriculum goes into, talk about the grant, the collaboration that's led up to it, and then also talk about opportunities that we have with the grant to do faculty professional development, and finally finish with ways that we're continuing to enhance the program, bring in new material, and make sure that our students are engaged in kind of the full range of activities that are involved with it. So to start off with, to begin with, for everybody at college, just point out that this program meets what we are aiming to do at the college directly from the mission statement. We are here to put the needs and the desires and goals of students at the center of what we do, making sure that we have a group of students that are fully trained in a rapidly advancing technology like CRISPR gene editing is the best way to ensure that they're ready to enter into the local workforce. Very often it's difficult or very time consuming to bring new curriculum into courses, and the ATE project really allows us to be able to accelerate that process, and has worked out extremely well to do that. So Delaware Tech is the only community college in the state of Delaware. We have 4 campuses spread throughout the state. So we're Delaware, we're a small state, but we do have kind of represent the 3 counties of Delaware. The Stanton campus is the campus that I'm located at. We also have those located further throughout the South, closest to the beach area for anybody who's been or wants to visit Delaware is the Owens campus located in Georgetown, Delaware. And we also have an urban campus located in Wilmington, Delaware, our George campus. For the purpose of this talk, when we bring in curriculum like this, we're doing this at a college-wide level. So the biological sciences and biotech programs that we have in the college are offered both at our Stanton campus as well as at the Owens campus, the Georgetown campus. And won't have much chance to talk about the curriculum and how it's been implemented, but part of the discussion that we've had in professional development activities with faculty are hurdles that arise with it. And so I'm happy to have follow-up conversations with anybody who may be looking to incorporate new curriculum into their courses and are concerned about doing that in a multi-college or within a community, within a state, the multi-college system. Okay, so briefly, we've got 2 different degree programs in the science department where I'm a faculty member of. We have our biological sciences degree program, AAS degree, which is our connected degree. Students take advantage of this through articulation agreements to transfer to institutions to complete their bachelor's, starting off as juniors. For us, most of our students that graduate take advantage of this by going to the University of Delaware or Delaware State University. We also have a technician pathway, which we abbreviate to BIT. And the goal of this is to train students to enter into the workforce. That program is smaller in the number of students that are served, but we are successful in giving students the skills that they need to go forward there. We're very proud of all the successes that our students have in moving forward and obtaining jobs. And this is just a list of where some of our most recent graduates have gone to and the companies they've been worked with, both in academic and industry standings. I just point out at the end, we're going to talk about the grant, and the collaboration that we have is with the Gene Editing Institute, a research institute which is located within the CRISPR healthcare system, the #1 provider of healthcare for the state of Delaware. And fortunately, because of the connection we have and the training that we have for students here, we've had several students now that have gained employment with the Gene Editing Institute, in addition to students that have had internships while they've been students at the college. Okay, so we've had 2 ATE projects focused in this area. One which ran from 2017 plus a no-cost extension year into 2020, 2021 ended last year, and a new grant that started in 2020, the summer of. This grant is a collaborative project between the college and the Gene Editing Institute at Christiana Care. This is a great collaboration because we are able to utilize experts in the field of gene editing to help inform and share information, knowledge, and techniques with us that we're able to bring into the classroom. They are the experts in this area, working on cutting edge areas, both the fundamental research of gene editing as well as development of therapeutic items and educational activities. And so in this collaboration, we benefit greatly from those their knowledge and the ability to share that. We've been able to support the incorporation of curriculum, laboratory experiments, plus laboratory equipment to do gene editing experiments in our biological sciences and biotechnician programs at the college on both of the campuses that offer those programs. And in addition to that, we've also are able to provide professional development workshops. These are targeted to community college faculty to help disseminate this information and make it easier to spread the incorporation of this powerful and rapidly evolving science across the country into programs that they're also looking to train their students. And so I'm going to talk a little bit about these over the remainder of the talk. Okay, so what does this look like in the courses that we have at Del Tech? So far, all of the major curricular aspects, classroom and laboratory, have been included in our genetics and molecular biology courses, which are capstone courses and are part of the curriculum for both the BIS and the BIT students. So all the biology students are able to receive and be trained in this area. This includes classroom background scientific material on gene editing, additional scientific skills, such as identifying scientific literature. I was mentioning as people were coming into the room at the beginning, I like to have the students go look up a research article, share the findings, and every year I go through and track the number of new CRISPR articles that have been published since the preceding year when it's been offered, and in a rapidly evolving field, that number has increased dramatically each time we've done that. Students also learn how to design CRISPR experiments, generating the materials, how to design guide RNAs, how to choose Cas nucleases that work best for themselves, et cetera, using online tools that are geared towards doing so. And we also have 3 different lab activities, which I'll talk about in more depth coming forward. So students have the opportunity to do CRISPR at multiple different levels. Getting that hands-on practice is obviously a key part of the training that we're doing. Recently, we've also enhanced this curriculum through use of an investigation of the bioethics and broader concerns related to CRISPR. I've had an opportunity to have students connect with local industry in seminar talks, as well as share information with bioethics in national conferences and receive feedback and information to help enhance the programs. And we also are including the opportunity for students to get more skills in bioinformatics, specifically in determining the success and the readouts at the sequence level of the gene editing experiments that they're doing. Over on the right-hand side, to just demonstrate visually the increase in infrastructure and equipment necessary. This is a room that was identified to be our tissue culture training room. And because of the benefit and the impact of the grant, we were able to equip it with the equipment and materials necessary to do mammalian cell gene editing, something that had been identified as a need by local industry, by our advisory boards for years, and which we're able to have students trained in using this equipment to do CRISPR gene editing and gain those skills that are desired by industry. So able to have a win-win situation with that. The lab activities that we're incorporating and have students involved with have updated and changed over time and can take a variety of shapes. So initially, we did CRISPR gene editing with a yeast system where there's a knockout of the AD2 gene. Once that's knocked out, the yeast colonies take on a red appearance. So a nice visual phenotypic change to view. Since that time, we've incorporated an experiment that was developed at the Gene Editing Institute with in vitro gene editing, where a plasmid is edited in a tube. We specifically are doing editing of the LAC-Z gene, and then after that editing has taken place, the plasmid is then transformed into E. coli, and we do a blue-white readout at that point. So we are not editing E. coli's chromosomal DNA. We are actually editing the plasmid in a tube separate and just using the bacterial as a readout. And that's been a highly effective training method. I'll also point out that for the curricular aspect, if you're teaching any lab talking about plasmids, doing blue-white screening with the LAC-Z gene, if you have general microbiology and the equipment necessary to do transformation and bacterial cultivation, you can do this type of readout. And curricularly, if you have difficulty making changes to proved activities, this is kind of an easy fit. Finally, I show the pictures of the tissue culture training room that we have. We have students that are working with mammalian cells that have a mutated version, an active form of enhanced GFP, and through CRISPR-directed editing, they're able to repair that gene to restore function. And I have a picture of the cells that this has been done with, and you can see an overlaid image of those corrected cells that are now glowing green, as well as to the students that were in the course the first year that we offered the training opportunities to them. So that's kind of how we apply it here. The benefits of CRISPR curriculum, I think that everybody that's here has heard of it or interested in it. It's something that definitely is a good hook for student engagement and interest as well. It's an activity that can fit, and curriculum that can fit in a variety of biology courses, probably most biology courses at some level, whether it is just the background kind of awareness information, the specifics of the activity, or if you can put in the curricular aspects as well. We even have an English faculty member that has brought in bioethics into their course. You know, we definitely have students that are getting it, not just in science courses, but in other courses throughout the college. So the professional development workshops. Through the period of both grants, we've had a couple of different formats. We've been able to run 4 kind of weekend 2-day workshops where we talk about curriculum, background of CRISPR, and a presentation given by staff from the Gene Editing Institute, as well as have participants come in and do the in vitro gene editing lab. The other nice thing about the lab is it has a very quick readout time. So participants can do the actual gene editing one day and then see the results of it, at least with the blue-white phenotype, on the second day. So it's always nice to have that immediate gratification on things working. We've run one 5-day workshop where participants get a more immersive experience with CRISPR gene editing. Again, we have more time to talk about the science, the advances, where the field is going. But participants in the 5-day workshop actually did all 3 of the gene editing labs. So they edited yeast, the mammalian cells, as well as doing the in vitro reaction. And for the 5-day workshop, the participants certainly were able to take more out of it because they were engaged with it for a longer period of time. The discussion around it was very exciting, and a lot of faculty left with ideas of how they could bring it in courses or work with undergraduate students in research and have those kind of mentored experiences for them to engage with CRISPR. Finally, over the past 2 years almost now, we've obviously been living under very different conditions. We've not been able to have people on site in order to do the training workshops. So instead, we've had 3 virtual workshops where we've been able to present the background material, a video of how to do the in vitro gene editing reaction, and have discussions about curriculum in order to keep moving. All told, you know, we've had a number of these seminars. We've connected with over 50 faculty members from 40 different community colleges across the country. And we are hopefully, with a change of conditions, going to be hosting more in-person workshops coming up shortly. And we're looking to have our next one in May towards the end or just after the semester. So hopefully, if this is of interest to you and you would like to come in through the grant, we have the ability to support travel and lodging, and the ability for you to come in and attend a workshop. So a little plug for coming to visit. Okay, some other quick curricular changes that we've been bringing about. I mentioned that we've enhanced bioinformatic training as well as bioethics, focus on bioethics with CRISPR. In the bioinformatics realm, the Gene Editing Institute, members of that have developed a online web-based program to analyze sequences that are the result of CRISPR editing activities. The program that they have is called Decoder. It has a very interesting story with a high school student that was highly involved working with the staff there to come up with it. It is available online, as you can see, with decoder.org. It works by just taking AB1 files from Sanger sequencing, and you can enter in parameters about where you were targeting for editing, the area you were going for, and the information that comes back tells you what type of editing occurred, what types of changes were there, the frequency of changes, etc. The benefit of the program is also if you have some mixed samples that are going in, it will actually untangle those, deconvolute the sequences, and allow you to get a broader picture of what's happening. So it's a really powerful tool that students get experience with and kind of gives them a full circle view of the experimentation and how to read the results from gene editing experiments. Under the area of broadening understanding and training in bioethics concerns, in February of 2020, we had Delaware Tech students, including 15 from our capstone courses, attend a local day presented at Christiana Care and with support and organization from Delaware Bio Association, our industry organization in the state of Delaware for bioscience companies, talking about all of the issues regarding development of CRISPR, the ethics of it, everything down to the regulatory aspects and the insurance aspects, who's going to pay for the techniques, et cetera. So those students benefited directly from being there, and we've included information from that into our curriculum. It also was a demonstration to the industry members that came of the training that the students are getting with us. Also, I saw that the evaluator from our grant, Drew Probst, is in the room today, and he has greatly helped to expand the scope and the dissemination of our activities. He gave a presentation at the AAC and U virtual conference and transforming STEM and higher education this past fall, and during kind of a think discussion, we're able to share thoughts about future evaluations of how best to gauge students and how they're learning towards bioethics, as well as be involved in the conversation with other individuals in a bioethics focus group, and made good connections that we'll be able to incorporate with our students going forward. Kind of winding down the end, I'll just say that this project has been extremely visible and well supported at the institutions. The support for the collaboration, the activities go all the way to the top of both Delaware Tech and Christiana Care, as highlighted in numerous publications within the state. Our grant was also highlighted as one of the ATE impact projects in the 2020-21 year. Additionally, the in vitro kit has been commercialized as part of a three-way project with the Gene Editing Institute, Delaware Tech, but the kit is manufactured by Rockland Immunochemicals. It is available directly from them, as well as through Carolina and VWR, so that to make it easier for individuals to bring that into their classroom. Over on the right is a picture from the experiment when we did this, when I did it with my class in the lab for the fall in our genetics course. And you can see from the readout the number of blue and white colonies. For students, it's really powerful to show that the blue ones do not always, they indicate that editing take place, and shows that gene editing is not 100% precise, and certainly a good awareness to have as we move forward with the new technology. Okay, winding down the very end, I would just like to point out that some of the broader educational pieces mentioned that the Gene Editing Institute has developed a series of videos and curricular information under the name Gene Editing 360. Very interested in DEI connections, bringing in the next generation of scientists and connecting it with the advances that are taking place here. A number of videos showing gene editing, showing members of the lab from diverse backgrounds, very successful women scientists, and other members to show kind of a path forward for that next generation of scientists at all levels. So to wrap up, this is the team that we have from Dell Tech. Another faculty member, Dr. Mike Boney, who teaches at the Owens campus, has helped with our instructional discussions in our on-site workshops. Eric Kopia, and the grant and head of the Gene Editing Institute. Kristen Pisarczyk is our science educator and a graduate of Delaware Tech that we're very proud of. She helps to bring the science to us. Joe Probst is our external evaluator from Old Dominion University. I have some contact information up there. We've got the chat, and hopefully I have given enough of a background to interest people to come and join us in Delaware for our upcoming workshops. So thanks.
Thanks, John. I'm sure people have a lot of questions for you, but I am asking everybody to hold off on your questions until later in our talk. But you can also